Genome-driven precision oncology has altered the landscape of multiple tumor types that include solid tumors and heme malignancies. Lung cancer and thyroid cancer now have become the poster child for precision oncology. So in this regulatory policy session, I was discussing a case study of selective RET inhibitor preclinical clinical development into registrational studies that led to the US FDA approval of two selective RET inhibitors, Selpercatinib and Pralsetinib. RET is one of the oncogene fusions seen in multiple cancers, and in 1985, RET was identified as an oncogene in papillary thyroid cancer. In 2012, it was identified as an oncogenic driver in non-small cell lung cancer. In 2017, we had the first in human clinical trials of two selective RET inhibitors entering clinic. And in 2020, approximately 3 years from entering first in human clinical trials, two selective RET inhibitors were FDA approved, so that these drugs can be accessible for all RET positive patients, non-small cell lung cancer and thyroid cancer in the real world. So let's take the case of Selpercatinib. After confirming preclinical activity and doing all the preclinical studies across multiple RET mutations and multiple fusions, we undertook the Libretto 001 study. That was a dose escalation study. The dose, our recommended phase 2 dose was 160 milligram twice daily, and based on this recommended phase 2 dose, the phase 2 expansion was designed, and what it showed was responses in RET-fusion positive non-small cell lung cancer. In the treatment-naive patient, the response rate was 85%, and the treatment relapsed refractory cohort, the overall response rate was 64%. Based on this data, Selpercatinib received the US FDA approval for RET-fusion positive non-small cell lung cancer. Similarly, with the compelling data for both RET-positive medullary thyroid cancer and RET-fusion positive thyroid cancer, again, phenomenal response rates. The drug also received US FDA approval for RET-positive medullary thyroid cancer and RET-fusion-positive all thyroid cancers, and this was a line agnostic approval. The second drug was Pralsetinib, or the Blue 667. Again, after confirming high kinome selectivity for RET in preclinical models, the phase 1 study, the ARROW study, was designed. The dose escalation confirmed the recommended phase 2 dose was 400 milligram once daily dosing. The expansion cohorts consisted of RET-fusion positive non-small cell lung cancer, RET-positive medullary thyroid cancer, RET-fusion positive other thyroid cancers, including papillary thyroid cancer, and a basket cohort of multiple RET-fusion positive malignancies. Again, the response rate in the treatment-naive cohort and treatment-refractory cohort in RET-fusion-positive non-small cell lung cancer and RET-positive medullary thyroid cancer and RET-fusion-positive thyroid cancer was compelling enough to warrant an accelerated FDA approval of Pralsetinib in these tumor types. Again, this was a line-agnostic approval. So what do we learn from designing these precision targets? Again, these are precision medicines. When we move on from the traditional paradigm of one-size-fits-all medicine, where we use a drug without a biomarker using a trial and error method, right now we are moving to the precision medicine era, where we want to offer the right drug to the right patient at the right time. So again, I had an extensive discussion in this regulatory policy session about defining and designing clinical trials for benefit in this precision oncology era. Thank you.